Errors in meiosis. Non-disjunction is the failure of chromosome pairs to separate, either during meiosis 1 or meiosis 2. Fertilization of an egg resulting from non-disjunction with a normal sperm results in a zygote with an abnormal chromosome number. As humans age, the odds of non-disjunction occurring increase. You can see the non-disjunction occurring in meiosis 1 increases greatly after about 33 onward, and in meiosis 2, particularly after age 40. In most cases, human offspring that develop from zygotes with incorrect numbers of chromosomes abort spontaneously. This is one reason for the large number of miscarriages that happen during the first trimester of pregnancy. There are two main types of alterations, aneuploidy and polyploidy. Aneuploidy occurs when a gamete that has undergone a faulty meiosis and has an abnormal number of chromosomes unites with a normal egg or sperm. The zygote formed will have an abnormal number of chromosomes. In a trisomy, the zygote has an extra copy of a chromosome. If the zygote is missing a chromosome, it is called a monosomy. Trisomy 21 is the most common chromosome number abnormality, with 3 copies of chromosome 21. It occurs in 1 out of 700 births. Incidence of Down syndrome increases with age of either parent. Down syndrome, the common name for trisomy 21, includes a wide variety of physical, mental, and disease susceptibility features. You can see from this karyotype of an individual with trisomy 21 that for chromosome 21, there is an extra copy. Aneuploidy in sex chromosomes. Unusual numbers of sex chromosomes, those that determine sex such as X or Y, do not upset the genetic balance as much as unusual numbers of autosomes, all the other chromosomes, perhaps due to the fact that the Y chromosome carries fewer genes. Abnormalities in sex chromosomes result in individuals with a variety of characteristics, the most seriously affecting fertility and intelligence. The greater the number of X chromosomes, the greater likelihood of developmental and/or intellectual disabilities. Sex chromosome abnormalities illustrate the role of the Y chromosomes in determining a person's sex. A single Y chromosome is enough to produce quote-unquote maleness, even in combination with a number of X chromosomes. The Y chromosome carries a gene, SRY, that sets in motion a program that induces male characteristics. An XY individual with a mutated SRY gene will develop the characteristics of a female. Human Y chromosome disorders. Klinefelter syndrome, or XXY. The extra X chromosome produces symptoms in a male that include reduced muscle mass, reduced body and facial hair, and lower production of testosterone. It may not be diagnosed until adulthood. Turner syndrome, or XO. A female is born with one of the X chromosomes missing or defective. Developmental and medical problems include short height, heart defects, and failure to develop ovaries. In a karyotype of the chromosomes of an individual with Klinefelter syndrome, you can see that there are 2 X chromosomes and 1 Y. Turner syndrome is an example of monosomy, where we have the loss of the second sex chromosome, where one of the X chromosomes is either missing or defective. Polyploidy occurs when an organism has extra full sets of chromosomes. The extra set of chromosomes is provided by one of the parents during fertilization. Polyploidy is normally fatal in humans. Most pregnancies result in miscarriage or the child dies soon after birth. Rarely, a person will survive to adulthood but suffer from developmental and physical disabilities. This is an example of triploidy in humans. There is an extra set of chromosomes for a total of 69. You can see there's a third chromosome here where we would normally expect only 2. This is different from trisomy, or we saw the example of Down syndrome where we just had one extra copy of one chromosome for a total of 47. In this case, there's an extra copy of every chromosome. Polyploidy is normal in plants and is sometimes necessary for completion of certain stages in the plant life cycle. However, polyploids are normally sterile but can be propagated vegetatively. Polyploid plants tend to be larger and better at succeeding in farm fields. Cavendish bananas grown in humid tropical climates account for 99% of the exports to developed countries. While their triploid nature makes them strong and desirable for consumption, they're sterile and propagated vegetatively with cuttings, producing clones of banana trees. This is a version of asexual reproduction. Since they do not reproduce by meiosis or fertilization, sexual reproduction, there's very little genetic diversity. The lack of genetic diversity makes them very reliable in terms of productivity, but also highly susceptible to extinction from disease. If the immune systems of all individuals is the same, a single pathogen that evades that immune system can wipe out the entire population. That's the case with the TR4 strain of Panama disease, a fungal pathogen that is spreading across the globe and threatening to wipe out the Cavendish. A genetically modified version of the Cavendish has been developed, yet is not widely distributed, but businesses are hesitant to distribute, market, and purchase genetically modified organisms, GMOs. The GMO version is parasite and pest resistant, but GMO bananas produce new selection pressure on the fungus and selects for varieties of Panama disease that can infect the Cavendish. Some worry that pollen from sexually producing GMOs will spread into native populations, fertilize them, and alter them. The Cavendish banana is a perfect example of the evolutionary advantage of sexual reproduction and genetic diversity in populations. Sexual versus asexual reproduction. The rate of evolution in sexual reproducing species may be higher than the rate of evolution in asexual reproducing species. Sexual reproduction is an advantage in rapidly changing environments, as diversity protects against extinction. Asexual reproduction is an advantage in stable environments due to its increased energy efficiency. The Cavendish banana is well suited for stable environments, but not when they rapidly change. Translocation. Translocation is an error that occurs when we get crossing over between non-homologous chromosomes. So crossing over between homologous chromosomes is normal and expected and produces genetic diversity. But if we have non-homologous chromosomes, for example here chromosome 4 and chromosome 20, where a piece of one chromosome breaks off and attaches to another non-homologous chromosome, we can get these derivatives. That's a mix of two chromosomes, 4 and 20, where we get this derivative of chromosome 4 and derivative of chromosome 20 as a result of the translocation shown here. The location of genes relative to other DNA sequences is important, and translocations can have profound effects on gene expression. A translocation that occurs in humans between chromosomes 9, which you see here, normal, and chromosome 22, normal here, can result in a form of leukemia. The ABL gene from chromosome 9 here joins to the BCR gene on chromosome 22, which you see here in red, to form the BCR-ABL fusion gene, which you see here. The resulting changed chromosome 22 with this BCR-ABL fusion gene on it is called the Philadelphia chromosome. The BCR-ABL fusion gene is found in most patients with chronic myelogenous leukemia, or CML, and in some patients with acute lymphoblastic leukemia, ALL, or acute myelogenous leukemia, AML. Chromosome 18. Not all structural rearrangements of chromosomes produce non-viable, impaired, or infertile individuals. It seems an inversion of chromosome 18 contributed to the evolution of humans. This inversion is not present in chimpanzees, our closest genetic relative. It appears it occurred after early humans diverged from our common ancestor with chimps. Inversion occurs when a chromosome segment breaks from the chromosome, reverses its orientation, and then reattaches in the original position. This is seen here with chromosome 18. So we take this section and we reverse the orientation. So this, what I'll call the top, is flipped, so it's now on the bottom, and this bottom is flipped, so it's now on the top. So you can see the darker portion is now down here. The lighter portion is now up here. In the region of this inversion, there are two genes, ROC1 and USP14. These two genes are adjacent to each other in chimps, but in humans, they are more distant, suggesting a breakpoint occurring between these two genes. Also, humans and chimpanzees express USP14 at different levels in different cell types. This inversion may have led to a change in their expression levels in a useful way. ROC1 and USP14 encode cellular enzymes. Therefore, a change in their expression could alter cellular function in cells. It is not yet known how this contributed to hominid evolution, but appears to be a significant factor in the divergence of humans from other primates.